नमस्कार मित्रो, आपण पाठीमागच्या व्हिडिओजमध्ये प्रिन्सिपल्स ऑफ इनहेरिटन्स अँड व्हेरिएशन हा चॅप्टर सुरू केलेला आहे. त्याच्यामध्ये आपण मेंडेल्स फर्स्ट लॉ, सेकंड लॉ अँड थर्ड लॉ याचं डिस्कशन केलेलं आहे. त्याचप्रमाणे मॉडिफिकेशन ऑफ मेंडेल्स रेशिओज, दॅट इज अ न्यू मेंडेलिझम, याचं पण आपण डिस्कशन केलेलं आहे. फेअर वी हॅव सीन इंटरॅक्शन ऑफ जीन्स. नाव we are proceeding towards the chromosomes. In the chromosomes, the first point is chromosomal theory of inheritance. Mendel published his work on inheritance of characters in 1866. Mendel, who has got the results from the hybridization experiments, these results were published in the year 1866, but for many years it remained unrecognized till 1900, because at that time the communication was not easy. Nowadays communication is very fast, but in those days communication was very difficult, and that's why this work was remained unrecognized till 1900. This was because of the communication. Secondly, the concept of genes as stable and discrete units, according to the Mendels, these are the factors, these are stable and discrete units, and these are these stable and discrete units, they are controlling the certain characters, expression of traits or expression of characters, and pair of alleles which did not blend, they did not mix with each other. This was not accepted by the workers of those days, by his contemporaries, means the scientists of those days. The Mendel's work was not recognized till 1900 because of Mendel's concept of genes or the factors as stable and discrete units that control the expression of traits, and pair of alleles which did not blend, they did not mix with each other. This concept was not accepted by other workers, other scientists of those days. Walter Sutton, along with the Theodore Boveri, in the 1903 studied the behavior of Mendel's factors and behavior of chromosomes at the time of meiosis. Here, after that, Sutton and Boveri studied the parallel behavior of Mendel's factors, or Mendel, which recognized these factors, behavior of these factors and behavior of chromosomes at the time of meiosis. This was studied by Sutton and Boveri in the 1903. Based on their observations, they have proposed the theory. They proposed a theory and chromosomal theory of inheritance. Sutton and Boveri, they proposed independently in the year 1902 the theory of chromosomes. This theory identifies chromosomes are the carriers of genetic material. According to Sutton and Boveri, or according to Sutton and Boveri's theory, or according to the chromosomal theory, chromosomes, these are the carriers of genetic material. You can see here, these are the chromosomes. These chromosomes carry these genes. The chromosomes carry these factors. Here you can see capital A, small a. These factors, these are transmitted from one generation to the next generation, and they are the carriers of genetic material. This theory states that chromosomes are present in pairs. You can see here, these chromosomes, one chromosome and second chromosome, these are present in the pairs. During the gamete formation, this cell is forming the gametes. You can see here, these chromosomes, these are separated, these are segregated, and one chromosome enters into one gamete. Here, you can see here, this chromosome goes to this gamete, this goes to this gamete, this goes to this gamete, and this goes to this gamete. During the process of meiosis, the chromosomes which were present in the pairs, they are segregated, these are separated, and one chromosome of a pair enters into one gamete. During the gametic formation, homologous chromosome pair segregate or separate, assort independently during the meiosis. Thus, each gamete contains only one chromosome of a pair. Here there are chromosomes are present in the pairs, but in the gamete you can see one chromosome of a pair enters into the one gamete. Nucleus of gamete contains chromosomes which carry all the hereditary traits. Here, the nucleus of the gamete, it contains the chromosomes, and these chromosomes are containing all the genes or all the hereditary characters or all the hereditary traits. Male and female gametes are the male gamete and female gamete, sperm and egg. They carry all the hereditary material or the hereditary character. Just now we have seen. They are the link between parents and offsprings. You can see here. This is the organism. During the process of meiosis, you can see that gamete formation occurs and one chromosome of the pair enters into the separate gamete. That is, these are separated from one another. Here you can see, suppose these are the male gametes and these are the female gametes. When this gamete and these they fuse together, you will see that this is A and this is small a. This chromosome number is restored. Here, this is the N number of the chromosome. There is a single set of chromosomes. And when male and female gametes they fuse together, there is the chromosome double, or you can say that diploid chromosome number, it is restored or it is maintained. The fusion of haploid gamete and haploid female gamete restored the diploid number of the chromosomes. In 1933, T.H. Morgan confirmed the results of Sutton and Boveri. Sutton and Boveri, they have proposed the chromosomal theory of inheritance, and their results, they were confirmed by T.H. Morgan in the year 1933. Then chromosomes. Chromosomes are filamentous bodies present in the nucleus of eukaryotic cell. Suppose this is a eukaryotic cell and this is the nucleus of eukaryotic cell. In the nucleus of eukaryotic cell, these chromosomes are present. Here, chromosomes, these are the filamentous bodies. These are present in the nucleus of eukaryotic cell. The term of chromosome, this has been originated from two words, chroma. Chroma means color, and soma or som means body. From these two words, chromosome word has been derived, and this chromosome word was coined for the first time by W. Waldeyer in the 1888. Term chromosome, it is derived from two words. Chroma means color, som means body, and this term chromosome, this was coined for the first time by W. Waldeyer in the 1888. The size of the chromosome is not constant in all the organisms. This size varies from organism to organism or chromosome to chromosome. Suppose in case of human being there are twice n is equal to 46. Here you can see that there are 23 pairs. All these 23 pairs, these are not having the same size. They vary, or their length or breadth, it varies from chromosome to chromosome, and the size also varies from organism to organism. The size of the chromosome varies from species to species. Each metaphasic chromosome in the metaphase stage, you can see that chromosomes are very clearly seen. These chromosomes are highly condensed, and this chromosome varies from 0.1 to 3.3 micrometers in length. Length of the chromosome is 0.1 to 3.3 micrometers. 5. Thickness is 0.2 to 2 micrometers. Thickness of the chromosome, it is 0.2 to 2 micrometers. So, but although these are the figures, they vary from species to species. Chromosomes are visible during cell division. Chromosomes are clearly observed during the metaphase stage because these are highly condensed, and these chromosomes, they can be visible during the cell division. Chromosomes are capable of replication. Chromosomes replicate. Replication that is duplication. They play an important role in heredity, transmission of characters from one generation to the next generation. These are responsible for mutation, change in the structure of gene, variation, differences among the individuals, and this leads to the evolution. So chromosomes are capable of replication, and chromosomes are playing a very important role in the heredity, mutation, variation, and evolution of eukaryotic species. Chemically, eukaryotic chromosomes are made up of chromosomes. If you see the chemistry of a chromosome, you will see that chromosomes are made up of DNA, histone protein, and non-histone proteins. Means these are the three important constituents of a chromosome. If you see the functions of chromosome, broadly, chromosomes are the carriers of heredity. They transmit. Suppose here there is A gene, here there is B gene. These genes, these are transferred from one generation to the next generation, and that means chromosomes are the carriers of heredity. Chromosomes are capable of replication, and they are playing a very important role in heredity, mutation, variation, and evolution. These chromosomes, they are having a role in heredity, means transmission of characters from one generation to the next generation. They are having genes or the factors in which mutation can occur. Because of the mutation, variation can occur. Variation is observed in the organism, and this variation is very important for the evolution. Thus, chromosomes are capable of replication, that is duplication, and they are playing a role in heredity, mutation, variation, and evolution. Chemically, if you see the chemistry of a chromosome, chemically, eukaryotic chromosome, it is made up of three important constituents: DNA, histone proteins, and non-histone proteins. Along with these, other structures are present, but these are the very important constituents present in the chromosome. If you see the function of a chromosome, chromosomes are carriers of heredity, carriers of genetic material. They carry the genes from one generation to the next generation. Here we know, suppose these are the chromosomes. Here A is present, here B is present, here capital A is present, capital B is present. These genes are transferred from one generation to the next generation. Thus, these chromosomes, these are the carriers of heredity. Then, number of chromosomes. The chromosome number is not constant. The chromosome number varies from organism to from species to species. The number of chromosomes is specific and constant for a particular species. If we consider the example of human being, twice n is equal to 46. This is a constant chromosome present in case of all the humans throughout the world. Means in Homo sapiens, in the human being, chromosome number is constant. If you see another organism, maize plant or other plant, in all these species, Gia maize or the maize, in this chromosome number is constant. The term of ploidy means degree of repetition of basic chromosome number. Suppose basic chromosome number, if you consider the human being, basic chromosome number is n is equal to 23. This is the basic chromosome number. In the diploid organism, twice n is equal to 46. Here you can see that ploidy means degree of repetition of basic number. This is the basic number. Degree of repetition means it has repeated 2 times. This is repeated 2 times here. If you see, sometimes there is thrice n, there is 4n. This is not present in case of human being, but for convenience, we are considering this. The basic number is 23. This basic number is repeated twice, 2 times. Here 3 times. Here 4 times. This is called as ploidy. The term ploidy means degree of repetition of basic number of the chromosome, that is x, twice x, thrice x, etc. If any chromosome number in a cell is exact multiple of the basic chromosome number, then it is called as euploid. Here you can see that this number is the exact multiple of this basic number. This is exact multiple of the basic number. Then we shall see in detail. Euploidy includes monoploid, or these are called haploids, diploids. In case of haploids or monoploid, there is a basic chromosome number. Suppose basic chromosome number is n is equal to 7. Means here 7 chromosomes are present. Means there is a single set of chromosomes. At this place, twice n means 14 chromosomes are present. Two sets of the chromosomes are present. Here 3 sets are present. Here 3 sets of the chromosomes are present. 7, 14, 21. If 3 sets are present, these are called as triploid, 4 set tetraploid, pentaploid, hexaploid, octaploid, etc. So euploidy, this chromosome number, if you consider this chromosome number, this is the exact multiple of this basic chromosome number. Means there are 3 sets of the chromosomes. This is called as euploidy. Euploidy includes monoploidy or haploidy, diploidy, here there is single set, diploidy 2 sets, triploidy 3 sets, tetraploidy 4 sets. This is called as euploidy. Tetraploidy 4 sets are the Fourier number of the chromosomes. This is euploidy. Then you consider aneuploidy. In case of aneuploidy, chromosome number, it is not exactly multiple of the basic number of the chromosomes. So basic number of the chromosomes is 7. Here this is called, suppose 7 is the basic number of the chromosomes. Then suppose 2 sets are present, chromosome number is 14. But there is no 14 chromosome, their chromosome number is not 14. But sometimes there are 15 chromosomes. Twice n is equal to 15. This basic number is not exactly multiple of the 7 basic number of the chromosomes. Here 1 chromosome is added. Means this is not multiplied by 7. Sometimes chromosome number, suppose twice n is equal to 13. This is not the multiple of the basic number of the chromosome. Here there is 1 less chromosome. Here there is 1 more chromosome. This is called as aneuploidy. Aneuploidy means chromosome number is not the multiple of basic number of the chromosomes. Aneuploidy, it is divided into 2 types, hypoploidy and hyperploidy. Then we shall see the meaning of this hypoploidy and hyperploidy. Basic number of the chromosomes, n is equal to 7, for example, you consider basic number of the chromosome. Twice n is equal to 14. But sometimes 1 chromosome number is less. If 1 chromosome is deleted, then you can see that twice n is equal to 13. Means 1 chromosome is deleted. Twice n is equal to 1. Means 1 chromosome is deleted. Means 1 chromosome is less. And that's why this is called as hypoploidy. If 2 chromosomes are deleted, this is called as nullisomy. If 1 chromosome is deleted, this is called as monosomy. If 2 chromosomes are deleted, this is called as nullisomy. Means chromosomes are less, and that's why this is called as hypoploidy. Then hyperploidy. Hyper means above, hypo means below or less. Hyper means more. In this case, chromosome number, suppose basic number of the chromosome is 7, twice n is equal to 14, and at this place chromosome number twice n is equal to 15. This 15 chromosome number is not the multiple of basic number. That's why this is aneuploidy. But here 1 chromosome is added, 1 chromosome is more, and that's why in this case this is called as hyperploidy. Suppose twice n is equal to 16. 2 chromosomes are added. Twice n is equal to 14. There must be 14. But 2 more chromosomes are added. That's why chromosome number is twice n is equal to 16. Means more chromosomes are present. This is called as hyperploidy. Then hyperploidy is divided into 2 types. trisomy and tetrasomy. Trisomy means twice n is equal to 1. Twice n is equal to 1. Twice n is equal to 14. One chromosome is added. This twice n is equal to 15. Means one chromosome is added, and that's why this is called as trisomy. Then we say why it is called trisomy. Here, suppose in case of twice n, one chromosome is represented 2 times. One chromosome is represented 2 times. Means there are 2 chromosomes of the same type. But if suppose one chromosome is added, means this particular chromosome will be represented 3 times. This will be represented 3 times. म्हणजे अगोदर असणारे 2 आणि नंतर ऍड झालेला 1, म्हणजे हा पर्टिक्युलर क्रोमोझोम 3 वेळा रिप्रेझेंट होतो. दॅट्स व्हाय दिस इज कॉल्ड ॲज ट्रायसोमी. त्याचप्रमाणे 2 क्रोमोझोम्स ऍड झाले, दिस इज रिप्रेझेंटेड 4 टाइम्स. सो दिस इज टेट्रासोमी, ट्रायसोमी अँड टेट्रासोमी. So in aneuploidy, chromosome number is not exactly multiple of the basic number of the chromosomes. This is of 2 types. Some chromosomes are deleted, some chromosomes are added. In case of hypoploidy or chromosomes are less, if one chromosome is missing, this is called as monosomy. 2 chromosomes are missing, this is called as nullisomy. In case of hyperploidy, more chromosomes are added. If one chromosome is added, this is called as trisomy. If 2 chromosomes are added, this is called as tetrasomy. When chromosome number is not the exact multiple of the haploid set, this is called aneuploidy. Just now we have seen. Aneuploidy is either addition or deletion of one or more chromosomes. Just now we have seen this. Then structure of chromosomes. Chromosomes are best visible under the under the microscope when the cell is in the metaphase stage. Prophase, metaphase, anaphase, telophase, these stages we have studied. In metaphase stage, chromosomes are highly condensed and these are clearly seen under the microscope. At metaphase stage, chromosomes are highly condensed. Typical chromosome consists of 2 chromatids and these are joined at the centromere. These are the 2 chromatids. This is a chromosome, single chromosome. These are the 2 chromatids. These chromatids are joined at this place. This is called as centromere. A typical chromosome consists of 2 chromatids. These are joined together at centromere, and centromere is also called as primary constriction. This is a centromere. This is a primary constriction. Primary constriction consists of a disc-shaped plate. Here there is a disc-shaped plate in the primary constriction, and this plate, disc-shaped plate, this is called as kinetochore. This is called as kinetochore. You remember that sometimes question is asked at which point, at which part spindle fibers are attached during the cell division. Spindle fibers are attached to the kinetochore. या कायनेटोकोरला स्पिंडल फायबर्स अटॅच झालेला असतात, त्यामुळे हे 2 क्रोमॅटिड्स दोन्ही पोलला ट्रान्सफर होतात. and these are moving towards the opposite poles. So spindle fibers are attached to the kinetochore during the cell division. Some few chromosomes have 1 or more constrictions called secondary constriction. आता ही आहे ते, this is called centromere or the primary constriction. Constriction म्हणजे खाच. या ठिकाणी दिसते ती सेकंडरी कॉन्स्ट्रिक्शन, ही दुसरी खाच, आणि ही सेकंडरी कॉन्स्ट्रिक्शन सेकंड. ही सेकंडरी कॉन्स्ट्रिक्शन सेकंड एक, सेकंडरी कॉन्स्ट्रिक्शन सेकंड ही दुसरी. ही सम और फ्यू क्रोमोसोम्स हैव 1 or 2 secondary constrictions. At secondary constriction first, न्यूक्लिओलस ही सेकंडरी कॉन्स्ट्रिक्शन, या ठिकाणी जी सेकंडरी कॉन्स्ट्रिक्शन दिसते ही कॉन्स्ट्रिक्शन फर्स्ट, आणि या ठिकाणी न्यूक्लिओलर ऑर्गनाइझर. At secondary constriction first, nucleolus becomes organized. म्हणजे या ठिकाणी न्यूक्लिओलस तयार होतो, याला म्हणायचं न्यूक्लिओलर ऑर्गनायझर. सेकंडरी कॉन्स्ट्रिक्शन फर्स्ट, दिस इज कॉल्ड एज न्यूक्लिओलर ऑर्गनायझर. Here a satellite body, this is called satellite body. It is attached at the constriction second. या ठिकाणी satellite body, ही जी दिसते this is a satellite body. This satellite body, it is attached to the secondary constriction. Each chromatid contains a long, unbranched, slender, highly coiled DNA. हा जो दिसतो तो कॉइल झालेला DNA. This is a highly coiled, double-stranded DNA starting from one end to other end. This is called as chromonemata. Here you can see double-stranded chromosome. Double-stranded DNA. This is extending from one end to the other end, and these are called chromonema or chromonemata. Chromatid consists of double-stranded DNA, just now I have told you, which extends from one end of the chromosome to the other. You can see here, chromosomes are filamentous bodies. The size or the size of the chromosome varies in different organisms, in the different species. We have seen that. There is a primary constriction. This is called as a centromere. Secondary constriction. There is a satellite body. Here there is a nucleolar organizer at the secondary constriction first. Depending upon the position of a centromere, there are different shapes of the chromosomes. Here you can see that if suppose this is a one chromosome, this is a centromere. If centromere is present at the center, then afterwards, suppose it is pulled during the cell division. It is pulled by the spindle fibers. It will take a shape like this. These are the spindle fibers. It takes a pea-like shape. If centromere is present at the center, during the cell division, during the metaphase, during the anaphase, this will look like pea-shaped. This is called as a metacentric chromosome. Centromere at the center taking a pea-like shape. If suppose centromere it is present near the tip, this is a centromere. This is present means two arms are equal. Here one arm of the chromosome is smaller, one arm of the chromosome is more longer. Here in this case you can see that centromere is present near the end. This is called as acrocentric. During this will take a shape like J. This will take a shape like J. This is called as acrocentric. This is called as acrocentric. One arm is smaller, one arm is shorter, and one arm is longer. Then submetacentric. Submetacentric means this is a metacentric. The centromere is present just away from the center. म्हणजे हे दोन्ही जे आर्म्स आहेत एक थोडा छोटा असतो आणि एक मोठा असतो, परंतु जवळ मीन्स इट इज प्रेझेंट नियर द सेंटर. इट इज सेंट्रोमेअर इज नॉट प्रेझेंट एक्झॅक्टली ॲट द सेंटर. एक्झॅक्टली ॲट द सेंटर आला तर त्याला आपण मेटासेंट्रिक म्हणतो. पण ह्या सेंटर पासून समजा या ठिकाणी सेंटर आहे, तर या सेंटरच्या जरा पुढे हा सेंट्रोमियर आहे. दिस इज कॉल्ड ॲज सबमेटासेंट्रिक. दिस इज कॉल्ड ॲज submetacentric, and this will take a shape like L. तर हा L सारखा दिसेल. During the anaphase stage, this will look like L. This is called as acrocentric. Then, if suppose centromere is present at the tip of the chromosome, म्हणजे याला एकच आर्म आहे, दुसरा आर्म नाही, हा सेंट्रोमियर आहे. Means centromere is present at the tip of the chromosome. This is called as telocentric. Metacentric, centromere at the center. Acrocentric, centromere near the tip. Submetacentric, centromere near the center. Telocentric, centromere is present at the tip of the center, and this will take a shape of I. This will look like I. These are the spindle fibers. This will look like I. So these are the different types of chromosomes on the basis of position of a centromere. Then sex chromosomes. Here in the organism, there are two types of chromosomes. Chromosomes which are containing the genes responsible for body characters. These are called as autosomes. And there are some chromosomes which are containing the genes which are determining the sex of the chromosomes. These are called as sex chromosomes. Autosomes and sex chromosomes. In the human being, there are 46 autosomes and 2 sex chromosomes. In the human being, there are 44 autosomes and there are 2 sex chromosomes. Thus, chromosome number is 46. Two types of chromosomes: autosomes determining the body characters, sex chromosomes which are responsible for determination of a sex. These are called as sex chromosomes. Sex chromosomes are also called as allosomes. Autosomes and allosomes. Human being and other mammals, they have X and Y as X chromosomes, sex chromosomes. In the human being and other mammals, there are 2 sex chromosomes, X chromosome and Y chromosome. This is the X chromosome. This is the Y chromosome. Here, X chromosome is straight. This is rod-like. It is longer. Y chromosome, it is shorter. X chromosome is longer, Y chromosome is shorter, and these chromosomes are straight and rod-like. X chromosome is metacentric. This is the X chromosome. This is the Y chromosome. X chromosome is metacentric. The centromere is present at the center. Means both the arms are equal in length. This is metacentric chromosome. Y chromosome is acrocentric. Acrocentric means the centromere is present near the tip. Means one arm is longer, one arm is very short. One arm is longer, one arm is very short. This is acrocentric. X chromosome metacentric, Y chromosome acrocentric. X chromosome has larger amount of DNA. This has larger amount of DNA material. Hence, this X chromosome is genetically active because it contains more amount of genetic material, that is DNA. Y chromosome has less amount of DNA and large amount of heterochromatin. This Y chromosome is genetically less active. This is more active because more genetic material, more DNA. This is less active because less genetic material, less active. Both X and Y chromosomes show homologous and non-homologous regions. We shall see the meaning of this word. Similar parts of the chromosome. Suppose here there is A gene, here there is B gene, here there is C gene. At this place, suppose there is A gene, at this place there is B gene, and at this place there is C gene. Means the genes are similar. In these two parts, genes are similar, A, B, C genes are present, and that's why these regions are called homologous regions. These are the homologous regions because these are having similar genes. Here you can see that the genes which are present here X, Y, Z. Here suppose there is alpha, for example. These are having different genes. These regions are having different genes. These mean these regions are not similar. That's why these are called non-homologous regions. Homologous region having the similar genes. Non-homologous regions, these are having dissimilar genes. Homologous regions show similar genes, while non-homologous regions show dissimilar genes. Crossing over takes place only between the homologous regions. म्हणजे आता क्रॉसिंग ओव्हर जर असेल तर या ठिकाणी होऊ शकतो, या रिजनमध्ये होतो. म्हणजे इकडचा जीन इकडे जातो, इकडचा जीन इकडे जातो. दिस इज कॉल्ड क्रॉसिंग ओव्हर. बट दिस ऑकर्स इन द होमोलोगस रिजन. क्रॉसिंग ओव्हर डज नॉट ऑकर इन द होमोलोगस रिजन. या होमोलोगस रिजनमध्ये क्रॉसिंग ओव्हर होत नाही. सो क्रॉसिंग ओव्हर ऑकर्स ओनली between the homologous regions of the X and Y chromosomes. Non-homologous regions of X chromosome. आता आपण पाहिलं हा नॉन होमोलॉगस रिजन, हा होमोलॉगस रिजन. नॉन होमोलॉगस रिजन ऑफ एक्स क्रोमोझोम इज लॉंगर. हा नॉन होमोलॉगस रिजन हा लॉंगर आहे अँड कंटेन्स मोर जीन्स दॅन दॅट ऑफ नॉन होमोलॉगस रिजन ऑफ वाय क्रोमोझोम. हा जास्त लांब असल्यामुळे जास्त जीन्स असतात, या ठिकाणी शॉर्ट असल्यामुळं कमी जीन्स आहे. एक्स लिंक्ड जीन्स आर प्रेझेंट ऑन नॉन होमोलॉगस रिजन ऑफ एक्स क्रोमोझोम. X linked genes, हे आपण पुढे पाहणार आहोत. X linked genes are present on, suppose there is an A gene. This A gene, it is present on non-homologous region of X chromosome, and Y linked genes are present on, suppose here Y gene. This gene is present on non-homologous region of Y chromosome. X linked genes on non-homologous region of X chromosome. Y linked genes on non-homologous region of Y chromosome. These are present. Here, just now we have seen the number of chromosomes, structure of chromosomes, then sex chromosomes, etc. मित्रों, आपणास हा व्हिडिओ आवडला असेल तर कृपया लाईक करा, शेअर करा आणि सबस्क्राईब करा. धन्यवाद.